Okay. Thank you very much. I'm really honored to be here today, and we have a list of great speakers throughout the day. But to be honest with you, probably the main reason why I'm here today is the fact that I worked right next to Feng Zhang for a year and a half. So when you have a colleague who's really hardworking and productive, be really nice to them because they may actually invite you for a talk someday. But you should also be nice to people who aren't as productive too. That's really a nice thing. Yeah. So Alice gave a great introduction to single-cell analysis and single-cell RNA-seq. And I was recently at the NIH single-cell meeting just a couple of weeks ago, and it's clear that there has been a great significant amount of progress towards single-cell analysis, both in transcripts and proteome. But right now, the sense I get is like much of the analysis is really limited to just several hundred to several thousands of single cells. Now, this really isn't a problem. Is there a laser pointer?
Thanks. Here's the other one.
Okay. And this really isn't a problem if you're studying a small number of cells for lineage tracing or so in simple model organisms such as C. elegans. But for many other problems such as tumor cell heterogeneity in mammalian organisms, or if you're interested in unbiased cell type classification of, let's say, human adult brain that contains up to over 100 billion cells, this could be current throughput. It really isn't adequate. So. Okay, so over the last 2 years, we've taken some big steps towards this goal of trying to understand single cell biology in an organismal level, and now we have fairly modestly high throughput method of analyzing multiple single cell transcriptome using some of the methods that Alice talked about earlier. Now these methods generally use methods that dissociate the tissue, and then using either micromanipulation or FACS sorting or Fluidigm's new technology of using microfluidics to compartmentalize various RNA amplification methods and for RNA-seq. And recently, Jim Eberwein from Penn published a really nice study using a photoactivatable decaging of RNA capture probes in situ, and when they can shine their laser on either single cells or a subcellular compartmental region that can actually capture a regionalized RNA, and they can amplify that and do RNA-seq. So our method here at PhysSeq, this is something I'm going to talk about for the rest of my talk. So all these methods are great. The main thing is that here and now Alex talked about how we can scale this up to over 10,000 of cells, but still 10,000 is relatively small compared to billions of cells that we have in the brain. And this acronym for this method is called TIVA. And this way, a photo-activated laser capture method is great, but it's probably not as scalable as we would like. So, and by the way, all these methods really fail to capture the spatial context of gene expression. So why should we care about spatial expression, spatial context? Well, we are interested in really understanding the cellular phenotype at an individual level, but if we don't actually care about the spatial context, that's like human geneticists saying that I only study genes, I don't care about the land or the history. I mean, you really have to understand the geography, the climate, natural resources these people are under, because genetics is a function of natural selection. The cells are exactly the same way too. So in A, you're seeing a diagram of tumor cell evolution, right? So it's not just a stochastic process. Cells actually, they talk to one another. They sometimes inhibit or promote each other's growth. Sometimes they compete with one another, and they sometimes cooperate. Now this behavior can be seen whether you're looking at a microorganism or amphibians. And understand the landscape is absolutely important for understanding individual phenotype. And this is probably the same with neurons or any other developmental systems as well. So how do we actually do this for things like transcriptome analysis? Now, transcript is important because it's one of the most fundamental ways for genetic information to get out there, outside the genome, right? So as you know, FISH, or fluorescent in situ hybridization, is the gold standard in terms of sensitivity and, depending on the method, also specificity. But they're notoriously low throughput, and there have been a lot of efforts to multiplex FISH, or RNA localization, in single cells. So it really started back in the early 2001 from Robert Singer's lab, who really pioneered this field. So he used multiple color localization barcoding to do profiling of small number of genes. And more recently, from Lan Kai's lab at Caltech, he came up with a color transition barcoding, or FISH sequencing, in which he uses super-resolution microscopy combined with sequential hybridization, stripping, to understand the identity of the transcripts. But other methods, you really are limited to maybe a few dozens or maybe 100 transcripts, not even close to thousands of genes that a cell may express. And even beyond that, these methods are perhaps almost too sensitive. So meaning that if you are looking at a really abundant genes, there's a physical factor of optical diffraction limit where you just cannot get accurate quantitative measurement of gene expression. And that a key example is GAPDH. I mean, if you see a slide of GAPDH single cell transcript, you just can't count them. And imagine doing that for 1,000 other transcripts. So now, I can't go into all the limitations of FISH in this talk, but there are these and other problems with FISH and other targeted sequencing methods or detection methods. So we really decided to start from scratch and think the fundamentals of the whole approach. So after 2 to 3 years of kind of struggling with this issue and trying to figure out what the true bottlenecks were, I really found to solve not just RNA-seq or RNA detection method, but really just fundamental key problems in all cell imaging, period, which is how do you count objects within your image? I mean, you have background fluorescence, you have objects of different sizes, and how do you even multiplex them? So some of the things that I focus on for the next several years are, okay, let's definitely do an unbiased whole transcriptome sampling. Number 2, now I'm interested in not just single cells, but a wide area, because most developmental biologists, you want to study the limb or the head or some large structures with a spatial pattern, right? So I don't want to be doing just super-resolution microscopy. So how do we actually do wide field resolution imaging while still being able to count individual molecules? So that was really important. And third, now, regardless of what you use for actual imaging, ultimately you all have the upper limit. What you can count discretely. So how do we actually, is there a method for a resolution-independent molecular quantification? All this means is that if you can sequence it, then you can have a quantitative sequencing data. So the big breakthrough came in 2010 when we were able to generate a matrix of cDNA molecules in situ within cells and tissues that are super bright. So it was compatible with any kind of 3D or 2D imaging. And that process really involved first fixing your cells and reverse transcribing your RNA in situ. And then we add this amino LDUTP to add amine backbone to your cDNA molecules so they can be irreversibly cross-linked to your protein matrix. We circularize it. It has a little adapter, so you can use this for RCA, rolling circle amplification. And during rolling circle amplification, you also add amino LDUTP. So now your cDNA amplicon has all these kind of lysine-like backbone, right? So you can now cross-link it. So after you cross-link it with this molecule, it becomes a transparent hydrogel that you can boil it, dry it, you can do anything to it, and it stays, stable. And which means that you can actually sequence for a very long time. So the key thing is that this is a random hexamer. If you do this method with a random hexamer but do not add RT enzyme, you don't get any amplification. This is just too short to circularize. But if you do have an reverse transcriptase, then you get an efficient amplification of the original template, meaning that whatever you sequence, it has a cDNA sequence in it. So if this is the amplicon generated in solution viewed using TEM, we've also repeated this experiment with super-resolution microscopy. And on average, these things are about 200 nanometers in diameter. and also they contain about 1,000 copies of the original cDNA template, meaning that compared to a single molecule detection method or a single molecule RNA FISH, which can use up to 20 fluorophores per molecule, we're about 50 times, if not 100 times brighter than those molecules. This is why we can actually do all kinds of 3D imaging at a molecular resolution. So this just shows you the improvement in our optical characteristics, which enables all kinds of microscopy. And this also shows you that we can cycle this, we can hybridize and strip for many, many cycles, and we can still retain really great signal-to-noise ratio. The value actually goes up because we did this manually, and by the time you get to a 60 cycle, you actually get really good. So this is an example of our RNA-seq library in situ generation in iPS cells. So using confocal microscopy, you can see that you have nucleolus here. You can actually see nuclear membrane. You can see cytoplasmic vesicles. Here is an example of nuclear membrane, nuclear speckles. This is a condensing chromatin. This is a metaphase chromatin, suggesting that once you have an amplicon, even despite the fact that you are doing all these washes, they're not just freely floating around. They're retaining their original spatial distribution. So we can do this in iPS cells, fibroblasts, mouse. This is the FFP section, so mouse embryo section, brain section from the Allen Brain Institute. This is a whole mount Drosophila embryo. Almost any sample, and I haven't shown brain organoids and several other samples. Almost any sample that we looked at, our method seems to work. So this is a movie of Z-stack going through an iPS cell. So you can see the nuclear membrane, nucleolus, and nuclear speckles. This is a very important thing that I want you to remember because there are some areas in the cell that we know are full of RNA that our method just cannot penetrate. It's probably because they're very tightly cross-linked. But you'll see later that this actually helps us to eliminate the kinds of RNA that we actually don't want in our data set. So this is a nucleolus. There are about 100 amplicons in the nucleolus in iPS cells. So serial quantitation beyond microscopy resolution. So how do we achieve this? So this is actually the first problem I solved like 3-4 years ago, which is that sequencing lets you to almost titrate the object density that you're going to count. How do you do that? That is because you can use a sequencing primer that extends by 1, and these are just random sequences here. So what you have then is you actually are sequencing all the molecules that's present, but then if you then strip that and extend the sequencing primer by 1, and you're detecting 1/4 of the density that's present here, you extend it by another, that's 1/16 of the original density here. So if you keep doing this, you can actually make a serial titration curve from the images that you have. So even if at this point you can't individually count objects accurately, you can extrapolate the molecular quantities for every single gene that you have. And another point that we made in our paper, which was recently published in Science, that normal way of counting objects, you have to depend on intensity-based image processing, meaning that you set your threshold based on known and your experiment-specific signal-to-noise ratio. But in our case, we don't have an intensity threshold. We just call sequences from every single pixel, no matter how dim they are, and the probability of them actually being a false positive is just purely related to how long your sequencing reads are. So we have a way. So by eliminating all kinds of intensity filter, we can identify objects purely based on its sequence information. And once we do that, we actually group related sequences into an object. So this is a different way of doing image analysis. And in the future, we think this is going to have a really big impact on how images are processed if you can tag your objects with the sequence. So we're currently, we just submitted a protocol paper. It should be, hopefully it'll come out within the next month or two. So our protocol, a lot of people think this is like a really hard protocol, but I think other RNA-seq technologies are tougher, except that they have automation and we don't. And we think we're going to solve that very soon. So The RNA-seq method, the library construction method takes 2 days, and this sequencing takes about 10 to 15 days. This is the hard part without automation. The actual image processing and base calling and data analysis takes 1 to 3 days, so it's not that bad. So just quick note about solid sequencing. We use solid sequencing instead of Illumina only because solid sequencing lets you to do this at room temperature. So we have our sample sitting in the microscope, right? We're changing all this fluid, and we just didn't want to build the flow cell around the microscope at 70 degrees. So that's the primary reason for us using this, and also the fact that solid sequencing chemistry, you can just buy it off the shelf. But we know that, so Illumina and both Live Technologies and Illumina are actually working with us right now, and Illumina chemistry works just fine. And the first demonstration of our method was shown by Reza Kalhor, another postdoc in our lab. He did essentially targeted the 5' end of the mCherry transcript, and he showed that we can actually sequence the 5' end of the transcript, sort of like 5' paste method. And if you add doxycycline, this is a doxycycline inducible transcript, so it comes up nicely. And then if you map where their transcripts lie, you about 98% map to the sense strand of the mCherry, and over 55% map to the within 2 base resolution of the transcript start site. So Evan Dory, he's a graduate student in our lab. He did bulk of the all the sequencing and base calling and writing all the softwares. So what he did is, just to rewind a little bit, so in order to do this manually, so you need a dedicated confocal microscope that nobody can touch for 2 weeks. And we had a really tough time finding it. And we finally found it, and we're incredibly happy, only to find out that its stage was broken. So we had an option of, okay, do we wait till the stages gets fixed, or do we just sequence this one field of view? And it was tough. It was a bitter pill to swallow, but we just focused on just one field of view. So this is from the experiment. So you do fibrilless imaging, and you do 30 cycles of sequencing, and you align all those images, and then and then for each pixel, you use bow tie, and then you map it to the reference library. And then you cluster those pixels into groups of objects, right? And then you say, okay, what are these objects? So vast majority, over 95%, map to the sense strand, so that's good. And about 43% were ribosomal RNA, which was lower than what we expected, but that actually enabled us to get a lot of mRNA reads. So what we were really surprised is that if you just, we got about 2,000 genes from that experiment, and then if you just take the top 100 genes and just look at it, you don't even have to go term analysis. What we found is like all of them were the fibroblast markers identified over the past 5 decades, just using various different types of experiments. And if you can take top 50 genes, 100 genes, or 1,000 genes, didn't matter what groups you pick, they just fall into collagen ossification, bone, skin developmental processes without doing any other bioinformatics. Now, when you compare this to RNA-seq, over genes that are observed more than 5 to 10 times, you have a correlation that's as good as 0.69 with fibroblasts, but with other cell types, the correlation is much lower, meaning that we really are detecting something that's functionally significant. And for every one of our pixels and object, we have an XY coordinate, right? So we can actually here's an image, and this looks like a nucleus, and this looks like a cytoplasm. We actually generate an image mask, right? And we say, okay, for every read, what is the relative enrichment in different cellular compartments? And what we see that is that mRNA, this is out of 4,000 genes, out of 4,000 genes, we're able to do this analysis about 200 genes. For these guys, they're mRNA and mitochondrial ribosomal RNA, which is in the cytoplasm, and non-coding RNA and other types of small RNA molecules. They're in the nucleus, such as MALAT1 and NEAT1. For very abundant genes with a lot of reads with 1.x5 coverage, we can actually detect physiologically relevant transcript splicing events. So I just want to drive this drive this point home, is that we compare this to 27 million reads of Illumina RNA-seq reads from the same cell line, and this is what you expect to see: housekeeping genes and things that are involved in just general cell functions and protein and RNA processing. This is why you have to do principal component analysis or relative fold enrichment bioinformatics. But if you were to take our data set, we only have 15,000 reads, and we can take either top 100, 500, or 1,000 genes. In this table, I use 1,000 genes. But immediately, all the things that you associate with fibroblasts, they pop up right to the top. So this told us, like, hey, we might be able to actually really get after the cell function just by using our technology without having to worry about what is our gold standard negative and positive controls and such. So again, we were also limited by the fact that we couldn't really scan a large area. So we just came up with a very simple a solution where we just grow the cell and we make a scratch and let the cells kind of crawl in. And we just say, okay, can we detect cells that are migrating in versus cells that are static, right? At least there should be some difference in cell cycle maybe or something, right? So when we did that, we did this for about 5 fields of view. And you can clearly see that there are some morphological differences, so that was good. So, and this experiment was done in the presence of EGF to mimic, the environment or the milieu present in actual wound healing, right? So as soon as we added EGF, what we found is that ribosomal RNA made up 80% of the transcriptome. Now, all the mRNA, they seem more or less, stable, but non-coding RNA, such as ribosomal RNA, they just jump right up. And this also suggests that using ribosomal RNA as your relative gene expression control may not be such a great idea. So again, because our reads are very informative, we can compare that with EGF. All the things that you associate with fibroblasts, they go down. Things that you associate with cell migration with EGF, they come up. So we, again, just like nuclear to cytoplasmic ratio analysis, we can actually create a mask for different regions of the dish. So these are all the reads obtained from the regions associated with cell migration, all the reads associated with contact-inhibited phenotypes. And then now these plots are noisier than they actually are because we use jitter plot to avoid overplotting. So what you see is that overall, the correlation between this region and this region is about 0.83. Now, one thing that I didn't show you, one slide that I skipped is that now we're growing the cell all simultaneously from the same batch. We change the media the same day, right? We fix it the same way, and we sequence it. You do everything exactly the same, right? So the technical noise or technical variation from region to region is a correlation like 0.9999. Because essentially, these are the same field, right? So if you're trying to take minor biological differences between 2 regions of the dish, we can really, our method is really sensitive. So if you do this, and there are about 12 genes that actually meet our arbitrarily defined threshold for what significance. So one's really obvious. The others are kind of on the borderline. But if you look at these 12 genes, 3 are related to collagen. But all eight of them are involved in actin, extracellular matrix receptor dynamics. They include GID4, PRPF40, and WINK1. So we don't think that this is an accident. So we think that PhysIC is able to identify genes de novo. So if you were to try to design FISH probes, you would never have targeted these genes because these genes are relatively fairly unknown, uncharacterized in the field of cell migration. Second, so this means that we can actually detect pathways de novo and discover unexpected players in players that are biologically important. So I just want to show you a couple movies. So this is a time series of sequencing reaction. You can just focus on one dot here, or this dot. So you have to realize that this is done over 2 weeks manually, right? In a room that's about 42 degrees hot because there is no ventilation, right? So our sample is incredibly robust and stable, right? And this is a view where the z-axis actually is time. So you're actually seeing 30 cycles of bases. So this bar represents a sequence barcode. So you're seeing a single cell with about 300 barcodes. We've gotten up to about 1,000 barcodes without a problem. But for this experiment, we weren't quite sure how well they would align for the paper, so we actually lowered the density a little bit. So you can almost think of this as a virtual nanostring technology. That's de novo. So what are our current priorities? Well, this whole process for each tile took us 30 to 40 minutes, right? So we're switching over to spinning this 3D microscopy, and we're seeing dramatic improvements of imaging speed. So using spinning this, we think, so we recently wrote a grant with Tony Zador and Ed Boyden. Our aim is to sequence 100 and then 1,000 brain sections first and second year. And with this gain in speed, we think that's totally doable. We also are about 2 to 3 weeks away from fully automating this, and this, the area that we can tile is probably going to be greater than 1,000-fold from our original experiment. So combined, our goal in the next 2 to 3 months is to really have a brain section or sections with over more than 10 million reads per section. So in our paper, we did not deplete ribosomal RNA because we actually want to use ribosomal reads as a control. Because they're so abundant, we can use that to estimate base calling accuracy and bias and all of that. But our next goal is to deplete ribosomal RNA. That should increase our per cell RNA count from 2 to 5-fold. That's going to actually allow us to get closer to single cell functional discrimination. So we're really excited with all these collaborations. They're all these are super excited collaborators. In fact, recently I started a Twitter account for the first time. I don't know how that works, but I have 4 followers: my wife, my sister, Ed Boyden, I don't know why, and Illumina. And Illumina just actually presented our work to the sequencing technology conference, meaning that I think they're pretty serious. Oh, I should mention that HiSeq 2000 is actually a spinning disk, right? That's fixed to just 2 planes, right? With automated tidal scanning. That's all it is. It's not magic. And what I just told you is I'm describing you how HiSeq works. So all we have to do is for all of you guys to tweet them and say, we need the machine tomorrow. And then I think they're going to be convinced that there's a need in the community. So again, with Tony Zador and Ed Boyden, we're doing massively parallel neuron cell type segmentation and transcriptome profiling in situ experiments. So Reza Kalhor just did a 33-round sequencing reaction on a brain tissue slice, and it looks fantastic, and hopefully he'll get his paper wrapped up very soon. Other things that include something I'm working on is you have a self-priming RNA barcode that's expressed within the nucleus that gets carried over and binds to an RNA binding domain, such as MS2 or N peptides from P22 and other viruses. And then you can actually tag individual transmembrane proteins with a specific RNA barcode. And so this is something actively under development. We're also collaborating with Peng Yin and other groups to really look at, and also Jim Eberwein, to really look at the distribution of transcriptome along the dendrite and exons. Robert Singer has recently published a paper in Science showing that RNA localizations in neurites are dependent on long-term potentiation. This is something we like to do transcriptome-wide. So you're actually seeing a bipolar neuron in vitro, and the red dots you're seeing is like amplicons present in all these neurites, and you can see that when you sequence them, they grow in 4 colors. So another thing I'm really excited about is I'm also investigating Wnt signaling for cell polarity in epithelial cells, in vivo and in vitro. So working with Hyun from the Wyss Institute, what we noticed is that all the epithelial cells that are grown in a flat dish, they just form monolayer. But if you put them in their native setting, which means that they have the apical side where they can diffuse their morphogens, and the basal side where they can also diffuse morphogens, they actually form villi in the case of intestinal epithelial cells. And in iPS cells, this is really exciting, is that they actually form inner cell mass right directly on the membrane. And we think that this extends to and we've also shown that using inhibitors like DKK1 or Wnt signaling, we know that these morphogens are involved in these in vitro morphogenic phenomena. But what we like to do is really to investigate transcriptome within this to see how transcriptome is distributed on the apical side and the basal side. And this is something that you cannot do with the current existing technology. So once things like DKK1 and Wnt1 and nodal and lefty, since they're all involved in breaking symmetry, now I think understanding cell symmetry and how you can actually break it by studying gene expression at a level, at a transcriptome level. So I actually think of this as chirality of gene expression, which is the ultimate kind of conceptualization of what we can do with a single cell resolution of spatial information of the transcripts. So getting back to my earlier slide, so I have projects that I'm starting to investigate, understanding tumor heterogeneity using a mouse model of pancreatic ductal carcinoma, and then tracking it and trying to define its transcriptome heterogeneity, as well as also to barcode them so that you can actually barcode them with either fluorophore or actual RNA expression barcode, and combine with single-cell RNA-seq that Alex talked about to really understand how the microenvironment contribute to either inhibition or promotion of certain cancer cell types. Obviously, if you were to combine this with targeted mutation sequencing as well as methylation sequencing, that'll be all the more powerful. So I'm not going to get into this, but I will say that we came up with this technology not because we are so excited about RNA-seq per se, but we think there's a really bottleneck in cell biology, meaning that next-gen sequencing is discovering thousands of mutations every day. Right? Anybody who does sequencing, they have molecular variants left and right. But we don't have the time to actually test them functionally. So how do we test them? So one thing that we thought early on, back in 2007, is that if we can have a library of modifying DNA, such as Cas9 library, or shRNA library, or cDNA library, infect the library of cells, and then have them undergo cell invasion or migration or any other assay that you'd like to do, can we actually just sequence a barcode and figure out which set of genes are responsible for a set of experimentally validated phenotype? So this is something that I'd like to pursue over the next 3-4 years. And this is my final slide, and this is kind of food for thought. Is that I told you about how our genetically encoded RNA binding domain could be really hooked up to almost anything you want, right? So you could think about neuroligand or synapsin or various other proteins. Not only that, now you're going to hear about genetically encoded voltage and calcium indicators, right? So if you had just picture in your mind, if you had all these blinking dots in live tissue sections, right, that indicate the activity of calcium level or something, right? And then you record the oscillations, you fix the cells, and then you sequence the barcode associated with each blink, right? And you think, oh, all these blinking dots in this region they belong to a same cell. And with our barcode, 30 bases will encode 10 to the 18th different cells uniquely. So you're not limited. So you can multiplex as many cells as you want. Now all you need is time to scan. So, all right. So this is my final slide. So I'd like to thank George. He came up with the idea of sequencing everything in situ back in 2003. He wanted to do in situ cell sequencing for a long time, but he first had to get the next-gen sequencing out of the way. And much of the work that you've seen here is done by Evan Doherty. He joined the lab about a year ago, or a year and a half ago, and really was absolutely critical in closing, getting our project to the finish line. And I'd like to also thank Richard Terry. He's been with the project for over 6 years, and he is very close to getting the entire process automated, which would be just phenomenal. And I'd like to also Allen Brain Institute and Kun Zhang. So both of these people really helped us early on just to think about why and where we might use our technology for. Okay, thank you.
So thanks for that. Let's take a couple questions, then we'll break for lunch. Yeah.
Do you have to modify the amount of linker that you put in if you're going to use Illumina? So you're switching from ligation to synthesis. Is it affected at all by the amount of cross-linking?
Right. So we've actually started playing with reducing the amount of cross-linkers. So we know that if you are really going after the single cell resolution where you want to see the nuclear membrane really nicely, right? Then we kind of tend to add more cross-linkers, but that does definitely inhibit our read length and also our sensitivity, right? But if you're interested in more of a kind of population ecology, right, tumor cell environment where single cell resolution isn't as important, right, then we actually really back off the cross-linker. And it looks like the protein environment itself, especially if you refix it, is enough to keep all the cDNAs in place.
Okay, another question? Yeah, Jeff.
If transcripts are close to each other,
Yeah.
Resolution becomes
Right.
an issue.
Right.
And close not only XY, but also Z.
Right.
And neurons.
Right.
So you just explain that a little bit?
Sure. So our method is like a really slow version of super resonant microscopy or STORM, right? Where they use stochastic blinking of subpopulation of particles, right? So we just exchange. So very simple, rudimentary way of doing what I showed earlier is like, let's say instead of one adapter sequence, right? Let's say you use 10 different adapter sequences connected to random hexamer, right? That means like if you just use one sequencing primer, you can subdivide your library pool into 1/10. So you get a much better spatial separation and resolution, right? So if you were to then use different sequencing primers, right, combine the result, so you had, you're counting every object, but each object you're getting a much better quantitative accuracy. And our method of just sliding the primer does that much more in an elegant way, where you don't need all 10, sequencing primers.
All right.
Okay.
So thank you. So let's give a big applause to all the morning speakers.